This video shows an animation of the TAC, the Tabletop Automation Center, performing a hypothetical clean room assembly of a medical diagnostic kit. The components of the kit include a cardstock bottom insert, which positions the other parts, a polymer-backed membrane test strip embedded with 3 lines of antibodies, a bottle of reagent fluid, a plastic suction tube, and a folded top insert, which is placed over the test strip. For the test, the patient uses the suction tube to place a drop of blood in the center hole of the top insert. Then the reagent fluid is poured into the cutout at the far right end. As the fluid moves through the membrane, the patient will see 1, 2, or 3 bands of color. A solitary band indicates a negative result. 2 or 3 bands indicate between different types of malaria. The components fit into a bar-coded box. A set of instructions is included. A lid is placed on the box and sealed with 2 pieces of tape. A time and date stamp is then printed on the lid. The process starts with a standard TAC equipped with 2 bi-level rotary magazines. The magazine on the left has a single motion, while the one on the right a dual motion. The standard gantry is replaced by an extra wide one, and the frame by a longer one. A custom pallet is placed on the lower disc of both magazines and loaded with boxes containing the bottom inserts. And the top left magazine is equipped with shelving to hold box lids. The carriage mounts 2 types of grippers and a small capping device. The right magazine holds more boxes on the lower disc. On the top disc are staged suction tubes, a folding table, and stacks of top insert flats. Also, bottle caps and folded and banded instruction sheets. On the work table is a cutting board with a fixture to hold the bottles, a sheet of membrane, a frame overlay, and pegs to locate the boxes. On the end of the TAC's extra long frame is added a forward gantry. On this forward gantry, the carriage is equipped with an ultrasonic cutting device, a rotary head device supporting 2 suction heads, and 3 inkjet spray dispensers. The first step of the process is to apply 3 lines of antibodies to the membrane. While the lines dry, the bottles are filled with reagent fluid by a dispenser working from the side. The dispenser has next to it an ultrasonic sensing device which checks to see if every bottle is properly filled. If the sensor finds anything amiss, then the TAC returns the bottle to the dispenser. The bottles are then capped. Next, the ultrasonic cutter is used to slice up lengths of the membrane. Keep in mind, the key to this entire application is to keep the membrane untouched by human hands. The TAC now brings boxes, one at a time, to the work table for loading. A barcode reader documents each box. A vision system establishes that a box is loaded and contains a folded bottom insert. Then a filled bottle, after having its barcode read, is placed in the box, followed by a suction tube. The work table turns to let the ultrasonic cutter finish cutting out a test strip. The suction cups on the rotary head then convey the test strip to the box. The carriage moves to pick up a pre-scored top insert flat from one of the stacks and places it on the vacuum folding table. The magazine then rotates to align the folding table with the wide grippers. The narrow grippers bring the top insert to the box. Please note, the actions in these animations are usually shown slowly in a step-by-step process to clarify what's happening. In actual applications, the tack would be moving faster and making multiple motions simultaneously, speeding things up considerably. The vision system now checks the location of the bottle, suction tube, test strip, and top insert. A set of banded instructions is then placed in the box. Which requires another vision check. A lid is then placed on the box. The assembled box is then returned to the magazine from which it came. This completes what could be considered the semi-automatic version of this application. All the assembled boxes would be located on the bottoms of the two magazines. Operators could then tape them up and run them under a print head to date them. Or, alternatively, a second tack could be used to complete these steps in a more fully automatic system. In this more automated system, a second tack would be employed, fitted with a single gantry supporting a print head, a rotary head device, and a tape sensing device. The work area would have a box holding fixture and a stand to hold an automatic tape dispenser. The second tack's one magazine would be fitted with another customized pallet. The carriage would take boxes in groups of 4 to the work table. Note, in an actual application, the second tack would begin operating once the first tack had started delivering assembled boxes on the second magazine. The first tack would then continue assembling boxes until they were all delivered to the second tack. The work table, in turn, delivers the ends of the boxes to the tape dispenser. The sensor verifies that tape was applied to the 4 boxes. The boxes are turned around by a gripper mounted on a rotary head to tape up the other ends of the boxes. The new taped ends are verified by the sensor. The right head prints the time and date on the tops of the boxes. The boxes are then stacked on the output magazine. Eventually, all the boxes are finished and placed on the output magazine, completing this, the more automated version of the assembly process.